In this video, we'll be talking about the platelets. And because the platelets are the clotting specialists in our blood, we'll also be talking about hemostasis. The prefix hemo, of course, refers to the blood, and stasis refers to a period of inactivity or equilibrium, sort of a standing still. So hemostasis is basically stopping blood loss. In the image over here on your left, you're looking at platelets in action. These are activated platelets, so they've been turned on, and they are making a blood clot. So you can see the platelets are all kind of bunched up in these groups, and they have these stringy fibrin tendrils kind of clumping them together and holding them together. Fibrin is the protein that's a major component of the blood clot. On the picture on the right here, you're looking at platelets and red blood cells, of course, in a normal blood smear. The platelets are these little tiny purple dots. Notice that the platelets are a lot smaller than red blood cells, and that's because the platelets aren't actually cells. What they are cell fragments. The cells that make platelets are called megakaryocytes, and these megakaryocytes basically break into pieces, forming platelets. Megakaryocytes form from the hematopoietic stem cell, the hemocytoblast, same place that red blood cells and many of the leukocytes come from. But one subset, one pathway that comes off from this hematopoietic stem cell includes a precursor cell that undergoes multiple rounds of mitosis without actually undergoing cytokinesis. Remember, mitosis is technically the process of the nucleus dividing, and cytokinesis is division of the cytoplasm. So if you have mitosis without cytokinesis, the nucleus and the cytoplasm just grow and get bigger and bigger and bigger until eventually you have the mature megakaryocyte. And it's this megakaryocyte that fragments into the platelets. So the platelets are really just little pieces of cytoplasm from the megakaryocyte. There's a brief video of this happening that I want to show you.
Megakaryocytes are the precursor cells from which blood platelets derive. These gigantic cells undergo an elaborate fragmentation process that pinches off portions of the cell's cytoplasm. These fragments are the platelets, which are then swept away in the bloodstream. Platelets are important for blood coagulation at sites of injury.
Platelet development is also called thrombopoiesis. And that comes from the other name for platelets, which is thrombocytes. That name comes from the fact that the term thrombus is another term for a blood clot. Now, let's talk a little bit about the very important process that platelets play a crucial part in, hemostasis. Hemostasis is stopping blood loss. And hemostasis is a continuous process, but it can really be broken down into 3 stages. The first stage is vascular spasm. And vascular spasm is immediate constriction of a damaged blood vessel. Vascular spasm is just a constriction in blood vessel diameter at the site of injury. This constriction can last for 30 minutes or more and extends for several centimeters around the damaged site. With decreased diameter of the blood vessel, you have decreased blood flow to that area, and so less blood lost from the damaged site. Decreased blood flow also concentrates the clotting factors at this point of injury, so clotting can happen much more rapidly. The second stage of hemostasis is formation of a platelet plug. So this is where our clotting specialists come in. When you have damage to a blood vessel, it exposes connective tissue lying underneath the vessel wall. Platelets adhere to the collagen fibers in this connective tissue. So they latch onto the collagen fibers. You also have damaged endothelial cells start releasing molecules that activate platelets. So once platelets get activated and start adhering to the collagen fibers that have been exposed now, the platelets themselves start releasing aggregating signals. So what you end up with is a positive feedback loop where you have activated platelets triggering the activation of even more platelets. That's shown over here on the left. You see platelets adhering to the damage to the collagen fibers, platelets getting activated, and some of the molecules that they release, some of the aggregating signals that they release are actually ADP, serotonin, thromboxane A2, and over here you can see what happens if you add just one of these aggregating signals to a tube of plasma. Remember, plasma, the fluid portion of blood. Here it's nice and cloudy. If you just add ADP to it, it starts to clump up, and that clumping is coming from platelets gathering together. The initial latching onto the collagen fibers is stabilized by a molecule called von Willebrand factor. Patients that have a genetic deficiency in von Willebrand factor have an impairment in their blood clotting process. It takes them longer to form clots and longer to stop bleeding when they have a cut of any level of severity. You may have heard that taking an aspirin a day is a good way to prevent strokes or heart attacks that can be caused by inappropriate blood clotting. And aspirin works as an anti-clotting agent because aspirin actually inhibits one of these aggregating signals. Aspirin inhibits thromboxane A2, so it short-circuits this platelet plug formation. If everything is proceeding normally and you appropriately and you have a normal clot formation proceeding, this positive feedback loop means that that can happen very, very quickly, and the platelet plug is complete in about a minute. The platelet plug is good for plugging small holes, but you have to get to a more stable solution if you have a larger amount of damage. And this brings us to our third stage of hemostasis, coagulation, or actual formation of a stable, stronger clot. In clot formation, you have the platelet plug being reinforced with a fibrin mesh. And this fibrin mesh just traps everything in it. You've got the fibrin as this web-like factor. You can see some small platelets trapped in the fibrin mesh, but you can also see some erythrocytes and even some white blood cells stuck in there. Now there's this whole cascade of cleavage events that lead up to formation of the actual blood clot, this cross-linked fibrin mesh that you're seeing there. We're going to start at the very end, start at the last few steps that happen before formation of the fibrin mesh, and then we'll go back and talk about how you get there. So in these last two phases of coagulation, they all depend on activation of an enzyme called prothrombin activator. Prothrombin activator is sometimes also called prothrombinase. It's an enzyme, so you know you've got to have that ase ending, right? Prothrombinase. Prothrombinase got its name. It's called prothrombin activator because it activates prothrombin. Very good. So prothrombinase takes prothrombin, which is a clotting factor circulating in the blood, and cleaves it, forming thrombin. Right? And then thrombin is another enzyme that takes fibrinogen, which is a soluble clotting factor circulating in the blood, and activates fibrinogen. It chops it up into fibrin. Notice fibrinogen is soluble. Fibrin is insoluble. So as the fibrin comes out of solution, it, together with factor VIII, forms this fibrin mesh that you see. Okay? So prothrombinase activates prothrombin, making thrombin. Thrombin activates fibrinogen to make fibrin, and fibrin and factor VIII form the fibrin mesh. Prothrombin activator, or prothrombinase, is the key here, and there are two ways to get there. The first is referred to as the intrinsic pathway. In the intrinsic pathway, well, first of all, it's called the intrinsic pathway because everything that's needed to get a blood clot is already there within the blood. It doesn't need anything outside of the blood to trigger this clotting practice. All it needs are negatively charged surfaces. And these negatively charged surfaces include collagen, activated platelets, even glass can trigger clotting via the intrinsic pathway. Now, the intrinsic pathway is great. You don't need any outside elements, but it's relatively slow. There's multiple steps involved. There are a lot of steps in that cleavage cascade, so it's a slower pathway. If you have a lot of tissue damage, very, very extensive tissue damage, you want to have a way to get a blood clot very fast, because otherwise you're going to lose a lot of blood, right? That's where the extrinsic pathway comes in. The extrinsic pathway is a very fast way to get to coagulation, but it requires a molecule called tissue factor. Tissue factor is found on the surface of cells that lie outside of the blood vessel, like muscle cells. So normally the blood is not exposed to tissue factor, but when you have extensive damage to a blood vessel, there's blood with its clotting factors is exposed to cells that have tissue factor on their surface, and this triggers a very rapid pathway to coagulation. This shows the more detailed cleavage cascades that lead to prothrombin activation. The intrinsic pathway is over here. And you notice how many more steps there are. There's a lot of cleavage steps, so it's a slower process. In the extrinsic pathway over here, if there's exposure to tissue factor, it's very rapid. Right? Notice that both of these pathways, they converge on factor 10. These are Roman numerals. So we have factor 12, and then active factor 12, factor 11, and active factor 11, factor 9, and active factor 9, factor 10, and active factor 10, which together with some calcium and some platelet factor and factor 5, activate prothrombin activator. There's a genetic disorder called hemophilia, meaning blood-loving, which is a bit ironic because it's a condition where people lose their blood. Hemophilia is a genetic disorder, and often it's due to a mutation in either factor 8 or factor 9, right? So if you lose one of these, you get hemophilia, and your blood can't clot properly. So you end up just bleeding profusely from even a very minor cuts. People with hemophilia need to get replacement factor 8 or factor 9, whichever one they're missing. So this chart shows you the overall picture of coagulation. Remember, we took this part down at the bottom first, and then went back up and looked at how we get to active prothrombin activator. And this is just putting it all together. So you have the intrinsic pathway to prothrombin activation, and you have the extrinsic pathway to prothrombin activator activation. And from there, it all proceeds similarly, with prothrombin activator activating thrombin, and thrombin activating fibrin, and fibrin forming your blood clot. Now, blood clots are great, and they stop blood loss, and they can stop blood loss for quite a while. They're longer lasting than a platelet plug, but they're still not a perfect, permanent solution. So there are a few things that have to happen in terms of repairing the damaged blood vessel. The first is you have clot retraction. Platelets actually have actin and myosin in them. So these are these protein elements that can kind of walk over each other, the same ones that you find in muscle cells. And so actin and myosin in the platelets contract, drawing the edges of the tear together. So they bring the damaged edges of the blood vessel closer together. You also have a growth factor that activated platelets released, PDGF, and that stimulates smooth muscle cells and fibroblasts to build that vessel wall back up. So smooth muscle cells will divide and rebuild the muscular portion of the vessel wall, and fibroblasts will divide and rebuild the connective tissue under the wall. Once you have the vessel wall rebuilt, you need to get rid of the clot, and this is where TPA comes in, tissue plasminogen activator. It's secreted by the endothelial cells, and it activates an enzyme called plasmin that initiates clot breakdown, or fibrinolysis. This enzyme is given to stroke patients and heart attack patients. It's as a clot-busting enzyme to break down clots blocking blood flow to the brain or to the heart. Now, excess clot formation can really be problematic. As we've mentioned, it can block blood flow in the brain, leading to a stroke. It can block blood flow in the heart, leading to a heart attack. It can block blood flow in the lungs, a pulmonary embolism. An embolism is just a blood clot that has broken away from the vessel wall and become lodged in a smaller vessel. So maybe it wasn't causing any problems where it was, but when it broke loose and got lodged in a capillary, now it's blocking blood flow, and now it's a problem. So controlling clot formation is really important. The endothelial cells that line your blood vessels help out with this controlling clot formation. They actually secrete molecules, nitric oxide and prostacyclin, that inhibit platelet aggregation. So if the blood vessel is fine, it's intact, it's undamaged, the endothelial cells are going to actively shut down platelet clumping. Normal blood flow also dilutes activated clotting factors. So normal blood flow, through undamaged, clean blood vessels, the clotting factors don't accumulate to the point where they start activating spontaneously. Also, there are natural anticoagulants in plasma. And basophils and mast cells produce heparin, a natural anticoagulant. It inhibits thrombin. Lastly, if you do get a clot forming, endothelial cells express a molecule called thrombomodulin. Thrombomodulin causes thrombin to activate protein C, which stimulates formation of plasmin, which, remember, is the enzyme that breaks down fibrin. So intact endothelial cells help out in controlling clot formation. Normal blood flow and natural anticoagulants in the blood limit clot formation. And basophils and mast cells help limit clot formation as well. As with most things when it comes to the human body, appropriate clotting is good, inappropriate clotting is a problem. And there are multiple mechanisms in place to inhibit inappropriate clotting. Proper nutrition is important for blood clotting. If you take a look back at the coagulation cascade, you'll see that calcium is very important. You have to have calcium ions in the blood in order to act as a cofactor for all of those cleavage steps. Vitamin K is also very important. Vitamin K is needed in order to make prothrombin and factors 7, 9, and 10. So the vitamin K comes in a number of different locations in order to actually make the clot. If you've ever used rat poison, or if you or someone you love are on blood thinners because they've had a heart attack or are at risk of a heart attack or a stroke, you may have heard of the molecule warfarin. Warfarin is used as a rat poison and also used in small doses as a blood thinner, clinically as a blood thinner. And warfarin interferes with vitamin K. That's how it inhibits blood clot formation. Alright, after studying this video, you should be able to talk a fair amount about what platelets are and how they're formed, and you should be able to explain how the body responds to vascular injury, including explaining the steps in hemostasis, so vascular spasm, platelet plug, and coagulation, and explaining coagulation.